Glycolysis. It is the metabolic pathway that converts glucose into pyruvate, pyruvic acid and hydrogen ion. The free energy released in this process is used to form the high energy molecules such as ATP and NADH. Two molecules of ATP and two molecules of NADH. Glycolysis is a sequence of 10 enzyme catalyzed reactions takes place in the cytoplasm. Glucose gets converted into glucose 6 phosphate and then to fructose 6 phosphate, fructose 1,6 bisphosphate and finally to pyruvate by series of enzyme catalyzed reactions. Next we have TCA cycle, that is tricarboxylic acid cycle. TCA cycle is also called as Krebs cycle and citric acid cycle. It is the second stage of cellular respiration. It occurs in the matrix of mitochondria. This metabolic process occurs in most plants, animals, fungi and many bacteria. But except bacteria, in all the other organisms, it takes place in mitochondria. This is the second stage of cellular respiration. TCA cycle consists of 8 steps catalyzed by 8 different enzymes. The TCA cycle is initiated when acetyl CoA reacts with the compound oxaloacetate to form citrate and to release coenzyme A. Then in a succession of reactions, citrate is rearranged to form isocitrate. Then isocitrate loses a molecule of carbon dioxide and then undergoes oxidation to form alpha ketoglutarate. Then alpha ketoglutarate loses a molecule of carbon dioxide and then it gets oxidized to form succinyl CoA, that is coenzyme A. Then the succinyl CoA is enzymatically converted to succinate and succinate is again oxidized to fumarate. Fumarate is hydrated to produce malate and at the end of the cycle malate is oxidized to oxaloacetate. Here each complete turn of the cycle results in the regeneration of oxaloacetate. So the end products of the TCA cycle will be 6 molecules of NADH, 2 molecules of ATPs and 2 molecules of FADH2. Next one we have fates of pyruvate. So the pyruvate is the end product of glycolysis, as you all know. This can be further metabolized in 3 different ways. Okay, that we say as fates of pyruvate. So here the key concept is considering these fates of pyruvate is the need for a continuous supply of NAD plus molecule for glycolysis process. As glucose is oxidized to pyruvate in glycolysis, NAD plus is reduced to NADH. It is significant here that each pathway of pyruvate metabolism includes provision for regeneration of NAD plus from NADH so that glycolysis can continue. Okay. Under aerobic conditions, pyruvate is oxidized to acetyl CoA. That reaction is catalyzed by pyruvate dehydrogenase. Then the so formed acetyl CoA enters into Krebs cycle. You can see in the image here. Then under anaerobic conditions or fermentation, pyruvate by glycolysis is further processed to give lactate or ethanol on carbon dioxide. When we ingest ethanol, it is metabolized by liver alcohol dehydrogenase. Here pyruvate is converted to oxaloacetate by pyruvate carboxylase enzyme. This is an irreversible reaction. Oxaloacetate acts as the substrate for the gluconeogenesis cycle. It occurs in the liver. Then we have lactic acid fermentation. It is the enzymatic anaerobic conversion of pyruvate to lactate. It is also it can be a reversible reaction. This reaction is catalyzed by the enzyme lactate dehydrogenase. Pyruvate converted into lactate by the action of lactate dehydrogenase in the presence of NADH plus H plus, that is hydrogen ion, and NAD plus is also formed. This occurs in muscle. Next we have photophosphorylation. What is photophosphorylation? The production of ATP by the light dependent reaction is called photophosphorylation. As it uses light as an energy source, photophosphorylation may be either cyclic process or non-cyclic process. That we say non-cyclic photophosphorylation and cyclic photophosphorylation. What is cyclic photophosphorylation? It involves the use of only one photosystem, that is photosystem 1, or we call it as PS1, and it does not involve the reduction of NADP molecule. When light is absorbed by photosystem 1, the excited electron may enter into an electron transport chain to produce ATP molecules. So following this, the de-energized electron returns to the photosystem by restoring its electron supply. Hence it is called cyclic reaction. The production of ATP by the light dependent reactions is called photophosphorylation, as it uses light as an energy source. As the electron returns to the photosystem, NADP is not reduced and water is not needed to replenish the electron supply. So electron returns to photosystem 1 by their incorporation to cytochrome. Only photosystem 1 is involved in the cyclic reaction. Next is non-cyclic photophosphorylation. It involves two photosystems, that is PS1 and PS2, photosystem 1 and photosystem 2, and it does not involve the reduction of NADP. When light is absorbed by photosystem 2, the excited electrons enter into an electron transport chain to produce ATP in this case. So, photoactivation of photosystem 1 results in the release of electrons, which further reduces NADP to form NADPH. The photolysis of water here releases electrons, which further replace those which are lost by photosystem 2. Here photosystem 1 electrons are replaced by photosystem 2. These are some of the differences between cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic reaction is associated with only photosystem 1, whereas non-cyclic, both photosystem 1 and 2 are involved. In cyclic, the electron expelled from chlorophyll molecule is cycled back, whereas in non-cyclic, the electrons are not cycled back, but they are compensated by the electrons from photolysis of water. In cyclic reaction, photolysis of water and evolution of oxygen doesn't take place. Photolysis of water and evolution of oxygen takes place in non-cyclic photophosphorylation. In cyclic photophosphorylation, phosphorylation takes place at two places, whereas in non-cyclic only at one place. NADP is not reduced in case of cyclic. NADP molecule is reduced to NADPH2 in case of non-cyclic photophosphorylation. Next we have electron transport chain. The electron transport chain is a series of protein complexes that couple redox reaction, that is reduction and oxidation reactions, by creating an electrochemical gradient that leads to the creation of ATP. Electrochemical gradient means the ions which are free to move through the membrane. It occurs in both cellular respiration and photosynthesis. First, the electrons come from breaking down organic molecules and energy is released in electron transport chain. Later, the electrons enter the chain after being excited by light, and then the energy is released and it is used to build carbohydrates. So it occurs in mitochondria. You can see in the picture inner membrane of mitochondrial matrix. The electrons comes from the breaking down of organic molecules and energy is released from them. Then the electrons enter the chain after they gets excited by light energy, and the so formed energy is used to build carbohydrates.